Good morning. As we have said in the last session, that the chromosomes are made up of DNA, or the chemical components of the chromosome is called DNA. D refers to the deoxyribose sugar, M refers to that it is located in the nucleus, and A corresponds to the phosphoric acid, which is H2PO4. So the chromosome is made up of sugar, which is called deoxyribose sugar, and it's located in the nucleus, and also it contains an acid. We have said also that the DNA is made up of two-stranded, double-stranded. Each strand is made up of a backbone. This is the first strand, and this is the second strand. Okay, this strand is made up of a backbone. The backbone is made up of acid. This is the phosphoric acid, connected by deoxyribose sugar, which is abbreviated by D. Okay, so this is the first backbone of the first strand, and this is the second backbone of the second strand. Okay, also we have a bond between deoxyribose sugar and a nitrogenous base, which are the adenine, thymine, guanine, or cytosine. So we have four nitrogenous bases, which are abbreviated by ATGC. Each nitrogenous base is connected to one deoxyribose sugar from the first strand. Suppose that we have C cytosine connected to this deoxyribose sugar, so C must also have a bond between it and between another nitrogenous base from the other strand. Right. Also, we have said that we have complementary base pair. What's the meaning of complementary? It means that every cytosine and nitrogenous base C is connected to guanine. So C is connected to guanine and A is connected to thymine. So A and C are complement to each other, and C and G are complement to each other. G cannot be connected to T and cannot be connected to A. Okay, right. The two strands chains are twisted over each other in a helical spiral form to make a double-stranded helix. The first strand and the second strand will make a helical spiral, يعني will twist over each other. بدهم يبرموا على بعضهم. Okay, like this, in this way. So we have this is the first strand. This is the first strand. It's coiled over the second strand. Okay, they will be in this form. Okay, you have to know that also that before recording we have to know that each double-stranded DNA means that we are talking about one chromatid. Yeah. In the mitosis, we have just one chromatid, then it enters interphase to become duplicated, right? This one chromatid, هيدي one chromatid هي عبارة عن double helix or double helical twisted DNA. Okay, so the double helix DNA, the two strands of DNA هن عبارة عن one chromatid. Okay, if we will have double chromatid DNA, double chromatid chromosome made up of double chromatid, this means that this chromatid هي عبارة عن double strand DNA in this form. Okay, and this strand, the other strand, كمان also هو شو it is double stranded DNA. Let's remember that the chromatin undergoes DNA replication to become chromosomes. يعني الchromosome will not enter mitosis before it duplicates. So it duplicates first by entering the interphase to become, it is called first chromatin because it is decondensed, okay, decondensed the chromosome. Then it undergoes duplication in this way, then condensation, coil up and condenses into chromosomes. In this form it is called chromosome, okay, because it is made up of double chromatin and also it coil يعني double stranded DNA عاملين twisting over each other, okay. So double stranded DNA twists over each other. In this way it will become, it condenses, okay. So it's called chromosome. Duplication of chromosome, in other words it's called DNA replication. يعني لما الكروموزوم يكون monochromatic, then it will become two chromatids. This is called chromosome duplication. We know that it's called duplication. Then what happens inside this chromosome, inside this chromatid or chromosome which is made up of monochromatid to become duplicated? شو بيصير بقلب الكروموسوم inside this chromatid? What will happen in order to be duplicated? What will happen is DNA replication, okay. اللي بيصير بقلب الكروماتيد is called DNA replication. During DNA replication يعني نحن الكروموسوم is duplicating itself، نكون عم بداوم الحالة، بيكون was made up of one chromatid بيصير made up of two chromatids. Let's see the stages of this DNA replication. أول شيء the chromosome of one chromatid is obtained at the end of mitosis. We all know that at the end of mitosis بيصير في عندي two daughter cells, okay, two daughter cells made up of 46 chromosomes, each chromosome made up of one chromatid. This single chromatid consists of double stranded DNA. As we have said, the single chromatid, one chromatid is made up of double stranded DNA. يعني هيدي الكروماتيد هي عبارة عن هيدي أول DNA, أول strand, and this is the other strand. You can draw it as straight, okay. This is the first strand only before it coil up, okay, before it twists over each other. This is the first strand and this is the second strand. هيدول التو strands هيدا عبارة عن one chromatid. This chromatid decondenses to enter G1 of interphase. The interphase is made up of G1 S phase and G2. Before it enters, or when it enters the G1, it decondenses. Decondenses يعني بيرجع مثل ما كان، اوكي. بيكون عم يعمل condensation لحتى يفوت على الميتوسيس. At the end of mitosis بيصير decondensed، بيرجع مثل ما كان بالأول. It passes through S phase of interphase. لما نفوت على ال S phase, what will happen? The DNA will make replication. Where the DNA double stranded opens، يعني هون لما بيكون هيك بيصير twisting over each other, okay. Then when it enters the S phase هون بيرجعوا بيعملوا separation. Where the DNA double strands opens بيعملوا separation, untwisting. Each strand undergoes replication by DNA polymerase. DNA replication يعني we will see in this figure how is the replication done. This is the first chromosome, the first stage of chromosome, which is made up of one chromatid at the end of interphase, at the end of mitosis. Then while it enters G1, the double helix opens, okay, will open. It is preparing itself to open. هيدي أول strand. and this is the other strand. These two strands uncoil itself بيكونوا two strands بيصيروا بيعملوا uncoiling while it enters before it enters the S phase بيصير في عندي denaturation يعني الستراكشر بيخرب. اوكي denaturation يعني by أمرار denaturation بتصير by heat يعني unwinding untwisting by helicase enzyme. بيحطوا enzyme in order to unwind or untwist the double stranded DNA. شو بيصير هون؟ the double stranded DNA will open. this هيدي أول strand وهيدي تاني strand. These two strands will be separated from each other. Okay, then a new strand will be formed which is complementary to the old strand. يعني هيدي the strand, the violet strand is the new strand. هيدي the new strand بتكون complementary لهيدي old strand. Okay, similarly in this old strand بيصير في عندي complementary أو complement strand which will be formed instead of the old one. يعني بيصير في عندي two new strands, each one is complement to each of the old strands. Okay, then rewinding بيرجع بيعملوا rewinding each strand. يعني this old strand with the new strand will make winding. بيرجعوا بيعملوا coiling or over each other, and this new strand and the old one will coil over each other also. Then we will have a chromosome made up of two chromatids. This is the first chromatid, which is made up of double helix, double stranded DNA, and this is the other chromatid which is also made up of double stranded DNA. Each double strand, this double strand is made up of a new strand, هيدي violet, new strand, and an old strand. And also this chromatid is made up of an old strand and a new strand. Then it undergoes condensation at the end of interphase. يصير في عندي condensation لحتى يصير في عندي chromosome made up of two chromatids.